are a few announcements I'd like to make for you. They are to my right on the board. The first announcement is just to remind you that next week you have to collect a sample of plant material and bring it into the lab. Your GSIs are going to grade you on bringing in the sample, and they're going to grade the answer to this question. Now, some of you will collect the wrong material. I wouldn't worry about that. The whole point is for you to go through the exercise of trying to think about what a bryophyte is, to imagine what it is, where it might grow, and to use that thinking to answer your question. There's no right or wrong answer for this question. We want you to start thinking about it, and that's the purpose of having you to collect. And just take your sample, if you collect it over the weekend, and wrap it in some saran wrap or in a plastic bag, just something to keep it from drying out, and then bring it to the lab if you have a Monday lab or whatever day your lab is during the week. The second point I want to make is that we have an individual by the name of Mr. Terry Kern, who is a retired biology teacher from community colleges, and every year, and I think he's going to do it this year, he holds sort of a review session where he provides a different perspective on plants from what I have given you in lecture. And his comments are generally very useful for helping you study, especially the more difficult concepts that I present. And he'll give you test questions, he'll give you practice questions, and that will be every Friday at 9 o'clock following the lecture in the same room, 2015, that we normally will have office hours. And if the class gets to be very large, or his session gets to be very large, we'll find another room for it. But in the past, students have found it very useful. You don't have to go every time, but it's something that you want to keep in mind because it really, from what I've heard from students, for many of them, it really has helped them, especially for some of the more difficult concepts that we're going to discuss. Okay, so what I'd like to do today is to continue talking about the fungi, which I introduced you to last time, and then to move on to some other groups of organisms which are going to be part of the 5-week period. Last time we talked about the fungi, and I gave you a list of generalized characteristics of the fungi. And today, yes?
Can you increase your volume? The two classes can't hear you. I'll read it.
Okay, this guy is the volume guy. Thank you. Yeah, you're welcome. So what we're going to do is to continue talking about the generalized characteristics of the fungi. And I'd like to begin with the first characteristic, which we said is that the fungi are generally filamentous. But they can be single cells, and we gave you the example of yeast. Most fungi are terrestrial, generally terrestrial, which means they grow on the land. But there are some fungi which grow in water. They are a minority. And we're going to show you some examples of so-called water molds or water fungi in the lab so that you can see that they take on very similar forms, they have very similar characteristics, except for the fact that they grow in the water. Now, we said that the fungus consists of filaments, and we give a name to these filaments. A single filament is called a hypha. And if there are many filaments, we call them hyphae. We add an E onto it. And many hyphae make up a structure which we call a mycelium. So these are really the three major terms you have to know with regard to the fungal body: hypha, hyphae, and the mycelium. We also said, and we'll review it again now in more detail, that there's both asexual and sexual reproduction in the fungi. Asexual can be from a piece of the filament breaking off, or, as you'll see in lab, some fungi will actually make spores asexually. And we're going to show you examples of that today from slides, and also you'll have examples to see of this asexual reproduction via spores when you have your lab next week. Now what I'd like to do is to spend some time talking about sexual reproduction. Because the fungi have invented an entirely new sort of theme on their sexual reproduction, which is found nowhere else among the biota, among all the living organisms that I'm aware of. And let's begin with a single hypha, or hyphae, a single hypha, and we'll divide it into cells. So this is a hypha. And in this hypha, we're going to put a nucleus. And this will take you back to your high school biology. We're going to say that the DNA level is haploid. So if you're unfamiliar with this term, go back and look in the textbook to familiarize yourself with it. And we sometimes often designate haploid by calling it N. So these nuclei in this particular filament is N. Now, fungi can spend a great part of their life cycle, their lifetime, many years in the soil as N hyphae. During this time, they're secreting enzymes to the soil, they're digesting organic material, and they're growing happily. However, sexual reproduction can occur when this hypha here meets a sexually compatible hypha of a different type or a different strain. Now, in fungi, they don't have just two sexes like most organisms do. Fungi have many sexes, and we call these sexes mating strains. Mating strains. So, if compatible, mating strains meet, they fuse and begin the sexual reproduction cycle. And let's talk about how that's done. So let's put two hyphae, or two filaments, just as we've drawn before. And let's say that they are of different mating types or different strains. Two hyphae. And for our purposes, we're going to say that each of these cells has a nucleus. Each of these cells is N. These are both N. And for the discussion, what we want to do is to just distinguish them as being different mating types or different strains. So we'll say that these are pluses and these are minuses. So just so that we can talk about two different strains or two different mating types. Now, when the two types meet, as we said, if they're compatible, they will fuse. And you will produce a structure, which again is a filament, which has cells. And now this is the truly remarkable and different part of the fungal life cycle. In each of these cells, you have two nuclei. You have a plus in each cell, and you have a minus in each cell, coming from the original fusion. This condition, because the nuclei remain separate, we call this N plus N, to indicate that the DNA in each of the nuclei has remained separate, N plus N. And another term which we frequently use to describe these, and which is used in your textbook, is because there are two nuclei in each cell, to indicate the two nuclei or to indicate this nuclear condition, we call it di for two. And the Latin for, I think it's Latin, for nucleus is karyon. So we call this the dikaryon because it has two nuclei, N plus N, in each cell. This is contrasted with this condition here, the monokaryon, where we have mono meaning one nucleus per cell. So now we form this dikaryon. This is all taking place probably in the soil or in the rotten log, whatever it is the fungi are digesting. This dikaryon or dikaryotic state can persist for many years. The fungus can continue to absorb nutrients, continue to grow. Each of the new cells that it produces will have two nuclei, a plus and a minus in this case. And then we think as a result of probably a change in the environment, perhaps the food runs out in the soil, there is a trigger. And this trigger, so we'll put a change in the environment. It could be food running out. It could be a lack of water. Something has changed. As a result of the change in the environment, we now form what is known as the fruiting body. And I use this word fruiting. It's a term that's used, but you could also call it the reproductive body. And all of you have seen reproductive bodies, fruiting bodies. When you look at a mushroom, typical mushroom that you buy in the grocery store, this is a fruiting body. And if we take this mushroom and we cut it down the middle, and we could put it under the microscope, we would find that almost every place on this mushroom, and we'll talk about where the one exception is, every place on this mushroom, it is made up of dikaryotic hyphae. That is, each of these tissues are made up of many, many filaments, and the nuclear state of each of the filaments is N plus N. So we've made this fruiting body, which is mostly dikaryotic, as a result of some change, some signal that the environment has brought about. Now, the sexual part actually comes from the fact that on the fruiting body, there are so-called, and many of you have seen these, gills. And I'll show you some pictures of them in a moment. These are very fine tissue-like structures that most of you have seen. These gills are important because on the gills, the nuclei in each cell fuse to make a 2N or diploid nucleus. To make a 2N or diploid nucleus. At this point, when the diploid nucleus is made, we can have sexual recombination. And this is rapidly followed by meiosis, again, a term that you may have to refamiliarize yourself with in the book, to make N plus and N minus spores. So the 2N part of the life cycle is very brief. It takes place only on a very tiny region of the fruiting body, or in this case, the mushroom that you know of. And it is resulting then in meiosis after sexual recombination and the production of new spores. Now, these spores are very tiny, and in general, spores are distributed by the wind. This is the way fungal spores are distributed. But many fungi have developed different types of mechanisms for making sure their spores get transferred from where they are to a better environment. And I'd like to talk to you about briefly one of these types of mechanisms, and it's found in a fungus that some of you may have seen before called Pilobolus. Let me draw Pilobolus for you. It's a tiny fungus, maybe 5 to 10 millimeters in height. It grows on dung. That is the waste product of animals. So here is the dung. And now let's draw this 5 to 10 millimeter structure which makes up Pilobolus. It's relatively simple. It has a stalk. On top of the stalk, it has a swollen region. And on top of the swollen region, it has a spore mass. The other characteristic which you need to know about Pilobolus, which is involved in its dispersal of the spores or the spore mass, is that Pilobolus bends toward the light. That is, it is showing phototropism. Shows phototropism. Now let's discuss, now that you know that it has a couple of characteristics including growing on dung and showing phototropism, let's discuss how it disperses its spores. So we'll begin by having the dung here. Here is the Pilobolus now, with its inflated region and spore mass, and here is the light from this direction. So the very first thing that happens with the Pilobolus is that, number one, it bends to the light. Bends to the light. After bending to the light, we get an increase in pressure in the swollen region. An increase in pressure in the swollen region. This results in a discharge, a rapid discharge of the spore mass towards light. Rapid discharge of the spore mass towards the light. And the spore mass usually lands on grass. It is now eaten, the grass is eaten by some animal, maybe a cow, by some animal. And then the spore mass is released when the cow defecates. Meanwhile, the cow is, of course, walking a lot of places, and so this results in the distribution of the spores. The fungus has used the cow as the mechanism for transport of the spores to new areas. Now, my question to you is, why in this life cycle is it important that this fungus show phototropism, that it is phototropic? I want you to think about this question. Why do we have to have this phototropic response in order for this fungus to survive, for it to continue its life cycle? Finally, what I want to do, and I'm going to put the screen down, but don't turn the lights off yet, guys. Finally, what I want to do is to talk to you about an unusual association of fungi between fungus and other organisms. And this association, an association between fungi and other organisms. And these other organisms are photosynthetic organisms. This particular association between a fungus and a photosynthetic organism, which can be an alga, which we're going to talk about next, or a cyanobacterium, you know of as a lichen. Know of as a lichen. And up here in front, I have one of the many thousands of lichens which grow outside. This structure, which many of you have seen and felt before, and I hope some of you come up and feel it, this particular lichen which we have growing here consists of a symbiotic association between a fungus and one or more photosynthetic organisms. Now, who does what in this particular symbiotic association? So it's a symbiotic association. The fungus provides the structure. And it also absorbs nutrients and minerals from the outside. Whereas the alga, let's say this one has an alga in it, provides fixed carbon. Lichens are very successful organisms. They grow in some of the harshest environments on the Earth, in the Arctic, for example, because they essentially are an independent factory, able to get both minerals from the substrate and carbon, fixed carbon, via photosynthesis. Now, lichens are organisms which frequently come into areas as some of the first organisms that have been destroyed or the environment has been greatly upset. And examples of this might involve, for those of you who are from the Pacific Northwest, Mount St. Helens. When Mount St. Helens erupted, all of the land, all of the soil was covered with lava. Lichens were some of the first organisms which come in. They would land on the lava. They would begin to grow. They grow very slowly. They secrete acids, and as a result of secreting acids, they are important in the beginning of the formation of soil, of new dirt, which then allows other larger plants to come into the area and recolonize it. So they're very important in reestablishment of the plant communities, and then generally all communities, in areas that have suffered great disturbance. Secondly, lichens are very sensitive indicators of pollution. If you go to an area and you see no lichen growing, that's an indication that there's pollution in the atmosphere. And so you probably want to avoid spending a lot of time there. Alternatively, if you see a lot of lichens growing in an area, you can suggest to yourself that this is probably an indication that this is not a polluted area, since they are such sensitive indicators. Let me show you now a few slides of the fungi and of the different organisms which we have here. Now, this first slide is of a fungus which is producing spores, and these spores are being produced asexually. This is a fungus which you will see in the lab, and as the spores come off of the top structure, they are released and carried away by the wind. And so these are the spore masses here, and we'll look at them in a little more detail. Here you can see one of those masses, and you can see the individual spores which are coming off of the structure. Now here is a mushroom. This is the fruiting body. It is all dikaryotic hyphae except for under the gills, which I'll show you in a moment. And when the spores are produced in the gill region, they are then carried away by the wind. Notice a large mass of the mycelium is below the soil level. So when you take off the mushroom, you're really leaving behind most of the body of the plant still in the soil, or of the fungus still in the soil. Now, if we were to make a parallel cut to this surface over here, as will be seen in the next slide, you can see the gills. Here is the central stalk coming up. The gills are these structures over here, and on the gills is where we get the fusion of the two nuclei. And those two nuclei fuse, then we have meiosis occurring, so this is one of the gills, to make, in this particular case, plus and minus spores. And here you can see the fusion has already occurred, and we are producing, in this particular case, only two spores. One is plus, one is minus, in this particular example. And in this example here, I just want to point out to you the point where we get the fusion of the two nuclei is where the gills are located, and the rest of the mushroom is dikaryotic hyphae, at least the fruiting body that's above ground that you see. Here is Pilobolus, the organism which is phototropic, and it, as I said, is a tiny organism growing on dung. And in fact, the inflated region can also act as a lens to help to focus the light. The organism bends toward the light, and then it shoots its spores as a result of an increase in pressure. There also is a web page. The address is given in your syllabus, which actually shows you the discharge. It's quite remarkable. It's only about 10 or 15 seconds long, of the spore mass of the Pilobolus. Very fast, and I'd encourage you guys to take a look at that website. It's really pretty interesting. Finally, I just want to show you some pictures of lichens. Most of you are familiar with lichens. Here they are growing on rocks, and as a result, they will secrete acid and cause a breakdown of the material, and eventually that rock will be decayed. Lichens come in a wide variety of colors and shapes, and this is the last picture of the lichen. I just want to point it out to you. And these lichens then, again, consist of large numbers of organisms which involve both fungi and something which is photosynthetic. Okay, so these are the fungi, and I want you guys to realize you're going to see some of these things in the lab. Please spend some time looking at them. Now we're going to move on to organisms which are probably more familiar to people as plants and which differ from the fungi. The next group of organisms that we want to talk about, like the fungi, were grouped together as a big group of organisms by Linnaeus because they seem to share a lot of characteristics together. And these organisms Linnaeus called the algae. But as a result of more supposedly refined molecular characteristics, we now know that many of the organisms that were originally grouped with the algae are in fact should be put with separate groups and are not together, as we will draw on our eukaryote curve, with what we're going to call algae. And those groups which have been subdivided, or for our purposes divided the algae into smaller groups, are the following. The green algae, these are new groups created sort of as a result of more molecular characteristics, although other taxonomic characteristics came earlier. The brown algae, the red algae, the diatoms, and the dinoflagellates. These are organisms which are about 130,000 of these organisms, which were originally grouped together with the algae, but which we now place in separate groups on the eukaryote branch as a result of a better understanding of some of their molecular characteristics. Notice in some of these cases, and in fact when you see the diatoms and the dinoflagellates, that they are divided, or they can be divided on the basis of color, pigment, the green, the red, the browns. These are yellow down here. There are other ways of dividing the algae into different groups. For example, the presence or absence of flagella. Flagella, which are whip-like appendages used in locomotion. So you could divide the algae into groups based upon whether or not they produce flagella. Another characteristic, which I'll show you in a picture of in a moment, is whether or not the cell walls have some special feature. So we could also divide them on the composition of the cell wall. But for our purposes today, for the discussion that I'd like to give you, and because we have a large group of organisms to consider in a short time, I want to divide the algae in another sort of artificial way. That is, to divide them into organisms which are primarily or exclusively unicellular versus organisms that are multicellular. Into this first group, that is organisms that are primarily or exclusively unicellular, that is a single cell, we would place the diatoms and the dinoflagellates. These two groups of organisms, plus small invertebrates, make up the plankton in the ocean. They are the base of the food chain. So they are exceedingly important, and the health of the ocean is exceedingly important in order for plankton to be able to prosper because they represent the base of the food chain. Within this second group, we would place the browns, the reds, and the greens. The browns, as we will talk about a little bit more, are probably most familiar to you, coming for as many of you might from Northern California, because they make up the kelps. We'll talk a little bit more about them in a moment. So let's begin first by talking about the diatoms and the dinoflagellates. The diatoms and the dinoflagellates tend to grow near the surface of sort of still or standing water along the shores. It has been estimated that about 50, and some people say 80% of the atmosphere is regenerated as a result of the photosynthetic activities of the diatoms. It's a large amount. So here's another reason why we have to worry about the health of the oceans, because this is where a large percent of our atmosphere is regenerated. Now these two groups of organisms, the diatoms and the dinoflagellates, are they grow exceedingly fast, but in general, they are kept in equilibrium. They grow at the surface of the water with sunlight coming in, and they grow at a fairly good rate, but they don't overwhelm the system. However, at times when there is a storm or some disturbance on the land that washes into the ocean large amounts of nutrients or minerals, we develop what are known as algal blooms. Which are a result of an increase in nutrients. And this particularly concerns the dinoflagellates. They grow and they multiply very rapidly to make this bloom, this large number of cells. And you know of the common term for an algal bloom. The common term for an algal bloom is a red tide. And why are red tides considered so bad for humans? Why do we not want red tides? The dinoflagellates produce a toxin. And when they increase in concentration during an algal bloom and fish ingest the dinoflagellates, they die because they are poisoned by this toxin. Now, you would say, well, okay, so we won't eat the fish growing in an area, but why do they close beaches when we have an algal bloom or a red tide? The reason they close the beach when you have a red tide is because shellfish also ingest the dinoflagellates, but they're able to concentrate the toxin to a level which apparently does not hurt them or influence them. However, if people then harvest those shellfish and eat them, we can die. So beaches are closed, and they're especially closed to shellfish fishing as a result of a red tide because of the toxin which is concentrated in them. The second organism which makes up this group are the diatoms. If I were to pick out a group of organisms of all the plants that we talk about that I would study more, it would be the diatoms. They are a remarkable group because their cell wall contains silica, the substance that makes sand. And as a result of having silica in the cell wall takes on many, many beautiful shapes. They look like snowflakes, and I'll show you a picture of them in a moment. Now, as the diatoms grow, and having the silica wall, which makes them heavy, but also very strong, as they grow and die, the diatoms, because of their heaviness, settle to the bottom of the ocean. And they can make layers of hundreds of feet thick at the bottom of the ocean, which, when uplifted to the land, makes this material that many of you are familiar with, which is known as diatomaceous earth. Diatomaceous earth. Diatomaceous earth is made up of the shells, essentially, the outer walls of once living diatoms. It has extreme uses for humans. It is used as a mild abrasive in toothpaste, in silver polish. It is greatly used in filtration systems because of the small pores between these small cells or the walls of the small cells. So this is of considerable economic importance for humans, diatomaceous earth. The other group of organisms are the multicellular, the green, the red, and the brown. And we're going to talk about them in a moment. But what I wanted to mention to you, I'll show you some pictures in a moment, is that in the multicellular, you have specialization of cell types. Specialization of cell types. And an example of the specialization of cell type is up here in front, and I want to show you. This is a plant which is about 4 feet high, say 4 to 5 feet high. It is a kelp, and it has a stalk. And to anchor it, it's developed kind of like a root system, but we call it a foot or a holdfast. A holdfast. And then it might have a air bladder. And it would have blades. Blades. So here is an organism which is multicellular, in which the cells have begun to develop into various parts or types. And I have an example of one of these kelps, this particular algae here. The holdfast, or whatever is holding it down to the bottom of the ocean, is here. You can come up and look at it afterward. You can see the stem. You can see the blades, which are the photosynthetic portion of this alga. And then this one has small inflated portions, which are the air bladders, which help the alga position the blades near the top of the water so that it can photosynthesize. So this is an example of a multicellular organism which is considered part of the algae. Now, the green algae represent, as we'll talk about a little later on, an important group for understanding the evolution of plants. Off the coast of California, as I mentioned, the main multicellular algae are the kelps, the browns. They are found all the way up to the cold waters of the Arctic and to the Antarctic. The predominant multicellular algae in the tropics are the reds. Now, algae have important economic purposes in our lives. For those of you, again, from Asian cultures, algae is often consumed as a source of trace minerals and nutrients. It's used in various Asian foods, and it provides this rich source of nutrients that are concentrated and that otherwise probably would be absent from the diet. Many of you use products of algae every day. The compounds known as alginates are extracted from the algae and are used as thickeners or emulsifiers in many of the food and personal products that we use. For example, shaving cream has a thickness to it because it contains alginates. Whipped cream has a thickness to it because it has alginates in it. And a product of the algae, the multicellular algae that many of you have already used a lot, especially if you've worked in the lab, is agar, which is used to prepare the media in petri dishes. Now, what I would like to do is to show you some slides. Actually, what I'm going to do is, before I show you the slides, I want to talk to you a bit about the life cycle of the algae, because this is probably the most important portion of the lecture with regard to understanding them. The algae live in the ocean. And in order for you to appreciate the life cycle of the algae, I want to take you back to your life cycle, the life cycle of a person. And let's put on here a male and a female. And each of you is 2N. Each of us is 2N. And as a result of meiosis, the male produces an N sperm, and the female produces an N egg. We then get fertilization. which gives you the new 2N individual. Many of the algae in the ocean have this life cycle. But the algae, which eventually evolved and became what we are talking about as the plants growing outside, and I'll talk to you more about them as we get to the plants, developed an entirely new life cycle. And I want to show you what that life cycle is. We're going to start out again with the 2N individual. Just like humans, the 2N individual. We're now going to get, in this case, meiosis occurring for both of these arrows. And in this particular case, instead of producing a gamete, these meioses result in a spore being produced. Not a gamete, but a spore. This spore is N, just like the gamete, but it's not a gamete. And for the purposes, we'll just have this spore over here. We produce 2 spores. We could produce many spores. Now, let's just follow one of these spores. What does this spore become? This spore germinates. Let me show you what it germinates and becomes. The spore germinates and becomes a new plant. A new plant. This plant is N because it came from an N spore. And now, this is the really remarkable part about this life cycle. This new plant now makes gametes. And by mitosis, it makes gametes. And it can make both plus and minus. So for our discussion, let's say it's making both plus and minus gametes. Or you want to call them male and female. These gametes are now N because they came from this N plant. And now we get fertilization. To make a 2N embryo. A 2N embryo. Which then grows into a new 2N plant. So let's make a summary of some of the important aspects of this life cycle. The first and most important aspect of this new life cycle is that fertilization is delayed. It's delayed by the insertion of a new plant. This is a really important aspect. This new plant, which has been inserted, is N, and we call the new plant, we call it the gametophyte, or N plant, because it produces gametes. The new plant or gametophyte, which produces gametes. Now, the first plant, the 2N plant on the top, is producing spores. So we call the 2N plant, we call it the sporophyte, because it produces spores. So the life cycle of this algae have involved an alternation of 2 different generations. This is the great invention of the algae, of the green algae in particular, in which some of them have a life cycle which is like ours, that is, where there's no delay in fertilization. But a large number of them have now developed this delay in fertilization and have produced the second or haploid plant, which we call the gametophyte. And we're going to see this theme, this alternation of the gametophyte and the sporophyte for the life cycle to be complete, continuing up through all the rest of the organisms that we discuss in this class. But it originated in the algae. Now, a point which I want to make for you is that because the algae live in the ocean, because they are water plants, the way the gametes get together in these plants is they have flagella. They swim because the water provides them a medium which allows the gametes to get together. And we're going to keep in mind as we move up the evolutionary scale the mechanisms by which the gametes get together. Let me show you now some slides of the algae. Okay. Oops, sorry, going backwards. That won't do it. This first slide is of a unicellular alga, and again, you're going to be seeing many of these algae in the lab. Here you can see the two flagella on this particular unicellular alga. This is a way in which the algae moves through the water. They take on many different shapes. This looks like two airplanes crashing together. Here we have the large photosynthetic organ, and this particular alga has the nucleus in the middle. So this is a unicellular one. Now we begin to see algae taking on a form which is more than a unicellular, multicellular. And these multicellular forms can be small colonies, and you're going to see some colonial algae in the lab next week. Or the colonies can get to be quite large. This is a colony of an organism that you'll be looking at in the lab, and it can consist up to 60,000 cells. Colonial algae can also be filamentous, where we have one algal cell next to the other joined, and these filamentous algae then are considered colonial. They're not unicellular, they're considered to be multicellular. Here is an example now of a kelp, of a brown alga, which has become a multicellular organism, and we have differentiation of the various parts. We have the holdfast down at the bottom, which keeps it anchored. We have this sort of stem-like structure, which is called a stipe. You needn't really remember that. And then we have the blade. This one doesn't have an inflated structure that's very obvious, in which photosynthesis goes on. Now let me show you some pictures of diatoms, because these are, as I said, a favorite organism that I think would be worthwhile studying. The diatoms have as their distinguishing feature silica impregnated or incorporated into the cell wall. And in Victorian times, people would take these diatoms before there was YouTube and television and so forth, and they would have competitions where they would arrange diatoms on slides, and often symbolizing family histories. These are extremely beautiful, these diatoms, and you can see how they attracted people's attention. Again, what we're looking at is the silica wall of the diatom, and these are dead, and when these diatoms settle to the base of the ocean, when you put them in your silver polish or your toothpaste, you're looking at bits and pieces of these walls which are involved in giving it a grinding action. Here's another pattern that you can get from the diatoms. Very beautiful. Here are the dinoflagellates. These are what caused the red tide. These organisms look like sort of something from a science fiction movie. They produce this nerve toxin, which is extremely potent and which can lead to death in humans if we consume it. Finally, I mentioned to you that algae have a lot of uses, and I talked about agar and alginates. This is a picture of an algal pond growing in Australia. You can see how big they are. This is a house over here by this arrow. These ponds which are growing algae, the algae are extracted, and they are used for food coloring, for the coloring in medicines, and in various types of things in which we want to have an organic and not a synthetic material. So the algae play an important role economically in many aspects of our lives. Okay, have a nice weekend, and I'll see you on Monday.